Advances made in the past few years open up great hope and also terrifying possibilities. For some years now, humans have been discussing the potential to use pigs to grow human organs. What will an individual say if offered a life-saving heart transplant from a pig? All life on Earth uses DNA as its operating system. We now begin to understand how to develop entirely new life forms. Hi, welcome to the 39th episode of the Quintology Files. In this episode, we're going to look at how humans have progressed from repairing faulty genes to manufacturing completely new genes. In the previous episode, we discussed how life appeared to be like software code, an elaborate instruction set which compiled generates a self-replicating organism. Understanding how to repair faults occurring to our genes is leading us to the creation of new biological capabilities, and indeed to new life forms, potentially using a different operating system from DNA to develop entirely new versions of life. In the last episode, we discussed how combinations of amino acids form proteins, and in particular shapes which enable the performance of specified biological functions. Humanity has grasped that the operating system for life, at least on planet Earth, is DNA, and that DNA is chemical software. Advances made in the past few years open up great hope and also terrifying possibilities. Medical interventions to repair errors in human cell division, the main cause of debilitating illnesses and of course cancer, have been made much easier by the development of prime editing. Compared with a tool created from CRISPR-Cas9, which we discussed in the previous episode, prime editing makes a nick in one side of the double helix of DNA and then inserts new code at that site. Another protein then triggers a cut on the other side of the strand, and the cell repairs by copying the new code into the second strand of the helix, completing the edit. An article in Nature magazine, published in October 2019, reported how a team corrected mutations that caused inherited blood disorder, sickle cell anemia. In another case, they removed 4 extra DNA letters in a particular gene that causes Tay-Sachs disease, a rare condition that destroys children's nerves and usually proves fatal by the age of 5. Theoretically, with research and development resources, we can now develop software packages to repair any section of our DNA that is damaged. Time and funding is the only constraint to being able to retain perfect bodies as we grow older.
So are we seeing applications moving out of the research labs?
Yes, even 10 years ago, it was estimated that the value of products manufactured by genetically engineered life forms accounted for 2% of US GDP, close to $400 billion. This amount comprised broadly equal contributions from pharmaceuticals, crop seeds, and industrial products such as food additives, fragrances, and biofuels. Many players now offer bioengineering services. For example, how to make an organism more productive. Synthase a UK synthetic biology software company, reported one project where it had bioengineered a 200-fold increase in the rate at which bacteria produced the desired product in just 4 weeks. Another bioengineer, Ginkgo, working with the chemical giant Bayer, developed microbes to manufacture fertilizer inside a plant's root system. Other companies are designing programming logic into protein design by, for example, adding conditionality or AND gates, which is providing new, more precise ways of knocking out cancer cells. The potential from this is simply staggering. Nowadays, we are increasingly concerned that new antibiotics are being defeated by viruses in ever-shortening timeframes. Bioengineering offers the potential to confront viruses with genetic codes they've never seen before, making such cells immune. Some are working on changes to codons so radical that synthetic biology is being created which will operate in a parallel biosphere, based on the original but no longer in contact with it, populated by creatures which neither infect nor are infected by DNA-based viruses. Maybe one day we use such techniques to create a biology to colonize Mars, to avoid the risk of contaminating any pre-existing remnant of life there.
What about the use of DNA for COVID-19 vaccines?
Yeah, incredible examples. There was a breakthrough. We all recall the amazing new vaccines exploiting DNA-based techniques developed during 2020 to combat COVID-19. The new vaccines used techniques already under development to target specific types of cancer. Laboratory-designed messenger RNA was deployed to target very specific changes in human DNA to enable our immune systems to trigger specially designed immune reactions to destroy COVID-19 cells. Pharmacologists seized on this opportunity, realizing that instead of trying to manufacture proteins with their complex irregular shapes, which are designed to fit into target cells to allow drugs to exercise their purpose, they identified a much more effective route, employing prime editing to deliver edits to our DNA to resolve a problem at source. Instead of trying to deal with the effects of a disease, in essence, a move from treating effects to repairing the cause. Probably the greatest discovery in molecular biology, following our discovery of DNA's double helix structure, is the realization that the rules of shape and sequence are linked. The shape of a protein depends on the intricate way in which the chain of amino acids which it consists of is folded. This folding follows the sequence in which the different amino acids are strung together on the chain. The sequence of the amino acids is a crucial part of the genetic information stored in the DNA sequence of the cell's genome. RNA is the key mechanism by which the stored data in DNA is activated. The sequence of amino acids determines the shape created, which in turn is crucial to performing its purpose. The code stored in DNA is copied into RNA and edited to form a molecule of messenger RNA. the messenger RNA creates a chain of amino acids as the message is read. Messenger RNA COVID vaccines developed by BioNTech and Moderna use this technique to enable messenger RNA to use ribosomes to create new amino acids which could recognize, attack, and destroy COVID-19 viruses. These vaccines comprised a messenger RNA sequence with a distinctive COVID spike protein wrapped in a liposome bubble, which when injected enters our cells. When ribosomes read the messenger RNA, they produce the spike protein and the COVID virus is attacked. Amazing. The capability of getting cells to churn out proteins which are not described by genes pre-existing in our DNA opens up a huge range of therapeutic opportunities. Drug makers are now moving from focusing on the shapes of proteins to studying sequence. Instead of targeting proteins that are already present, now they can target the processes which control which proteins get made in the first place, adding helpful new ones to the process and cutting out the production of harmful proteins.
So which kind of diseases can we expect the new messenger RNA to address?
Already, there are many new messenger RNA-based drugs in clinical trials for the treatment of cancer, herpes, lung disease, heart disease, and numerous inherited disorders, as well as brain diseases such as Alzheimer's and Parkinson's. The combination of sequence and shape carried by messenger RNA means that the once haphazard process of drug discovery dependent on matching the shape of small synthetic molecules to the nooks and crannies of the proteins they targeted can be systemized. A sequence which recognizes one gene can be switched out for a sequence tailored to a different gene. When the operation of a messenger RNA drug depends on its sequence, its target and action can be modified at the click of a mouse. Another stunning development during 2020 was the development by DeepMind, which is now part of Alphabet Inc., of software to predict the shape of newly invented proteins, i.e. proteins that do not occur naturally in Earth's biosphere. Predicting the shape of an artificially designed protein will enable us to gain some idea of how a new protein might perform and what use it might be put to. Such modelling power will dramatically increase our ability to design new proteins to solve specific biological problems. Given the potential number of new proteins that we can create from the existing range of amino acids, this opens up virtually infinite potential. Eventually, we might be able to reprogram our own DNA to continuously repair and renew all our organs. We might set out to enhance the capabilities of our organs, e.g., improve our eyesight, develop self-cleaning arteries to make ourselves more or less immortal. Clearly, humans now have harnessed the biology of life and are exploiting it for monetary gain. No one's really thinking about the boundaries. Animal testing of cosmetics on animals, including primates, generated a very negative reaction. But where will the line be drawn for synthetic biology? For some years now, humans have been discussing the potential to use pigs to grow human organs. We generally look down on pigs. Indeed, many regard pigs as impure. But what will an individual say if offered a life-saving heart transplant from a pig? More importantly, the DNA of a pig and a human share 96% commonality. A pig is actually quite closely related to us. How do we know whether it lacks a spirit or a soul? In the past, some Christians did so believe, including the Cathars of the Pyrenees in medieval times. All life on Earth uses DNA as its operating system. DNA created dinosaurs, which ruled the planet for around 300 million years, whilst we Homo sapiens sapiens have only been around for some 175,000 years. Researchers catalogued around 75,000 errors which have arisen in human DNA, and now we've discovered the tools which might be used to repair these errors. In May 2018, the New York Times reported on research by Geordie Paps of the University of Essex and Peter Holland of the University of Oxford that identified 55% of the genes in the human genome were found to have been present in the very earliest animal yet discovered, some 650 million years ago. This rather proves humans are the result of a very slow evolutionary process which has taken place on Earth over the last almost billion years. Evidence of this is plentiful. For example, the earliest sharks used the same gene sequence to make hemoglobin as humans do, indicating a common ancestor as the source of both dinosaurs, sharks, mammals, and eventually humans. So we have inherited how to make hemoglobin from the ancestors of the dinosaurs. Technically, our inheritance goes back even further. Both mammals and dinosaurs inherited identical code to manufacture hemoglobin from an even earlier common ancestor. Again, this points not to God creating mankind, but very much to God possibly designing DNA. Maybe not the specific life operating system we label as DNA, but the conceptual framework by which amino acids may spontaneously form, eventually string together to form genes, and thus single-cell, self-replicating organisms. The presence of specific chemical compounds and climatic conditions dictating what cellular functionality succeeds in adapting and thriving in that specific ecosphere. Thus, life form operating systems suitable for that specific ecosystem may evolve. In the case of planet Earth, our system is known as DNA. Each Goldilocks planet might evolve its own unique variant of a life form operating system. So, to recap, our knowledge has expanded from recognizing our biological operating system is in effect chemical software to understanding how to successfully repair genetic errors arising in cell division, how to bioengineer life forms to improve manufacturing production of foodstuffs, vaccines, etc. As we gain understanding of how the sequence of amino acids determines the shape and therefore the functionality of the proteins made, we see scope to design new biological functionality and graft such functionality into existing life forms. Indeed, we now begin to understand how to develop entirely new life forms. I hope you found this interesting, hopefully even thought-provoking. Please press the like button and please leave a comment. Thank you very much. See you again soon.